Hey, it's Dr. Taylor, undergraduate exercise science program director and associate teaching professor at the University of Kansas. And welcome to another edition of Fitness Facts. This is the second video in a two-part series where I'm answering the question, what does the term muscle memory mean? Today I will talk about muscle memory occurring in muscles. This form of muscle memory occurs outside of the brain and our muscles as a result of resistance training. This form of muscle memory alludes to the fact that it is much easier for an individual with prior resistance training experience to regain their lost strength and muscle mass after a period of detraining than it is for someone with no resistance training experience to improve strength and build new muscle mass in the first place. You may have heard a friend say something like, "I used to lift weights consistently for years all through high school and college. Then I quit weight training for a couple years, but my strength and muscles came back pretty fast when I started training again." So, how do your muscle cells remember being stronger and larger as a result of past training experience? And how does this memory allow you to regain strength and muscle mass more quickly when you resume training again after taking some time off from the gym? To understand this form of muscle memory, we first need to discuss some basic cell biology. Remember that muscle tissue consists of primarily water and protein. The instructions for producing muscle proteins are contained within various genes, which are just segments of DNA located in the nucleus of our cells. Muscle cells are multinucleated, meaning each cell has more than one nucleus. The multiple nuclei in a muscle cell are referred to collectively as myonuclei. Both animal and human studies suggest that genes inside the myonuclei of muscle cells operate differently after weeks and months of consistent resistance training. It has been hypothesized that muscle cells possess an epigenetic memory within the DNA of these myonuclei that enhances muscle strength and growth in a more rapid manner after the reintroduction of resistance training. The term epigenetics can be defined as changes in the activity and expression of genes brought about by structural modifications without altering the genetic code. Or more simply, epigenetics refers to changes in the ways that a gene functions, even though the gene itself is not permanently changed. The way a gene functions can be altered by attaching methyl groups to it, or by removing methyl groups from it. A methyl group is an organic molecule that consists of one carbon atom bonded to 3 hydrogen atoms. If a gene has an increased number of methyl groups attached to it, we call that hypermethylation. In contrast, if a gene has a reduced number of methyl groups attached to it, then hypomethylation has ensued. A 2018 study conducted by Seaborn et al. Provides evidence that human skeletal muscle cells possess an epigenetic memory of prior muscle growth, also known as muscle hypertrophy. In this study, untrained men performed 7 weeks of total body progressive resistance exercise with a training frequency of 3 days per week. This was followed by a 7-week detraining period in which no exercise was performed and muscle mass returned back towards baseline levels. The men then resumed the 7-week total body progressive resistance training protocol, with training sessions again occurring 3 times per week. Results from the study indicated that hypomethylation of several muscle hypertrophy-related genes occurred after the initial 7-week training period. Interestingly, this hypomethylation was maintained throughout the 7-week detraining period as muscle mass decreased. However, hypomethylation doubled following the 7 weeks of retraining. As a result of this increased hypomethylation, muscle hypertrophy-related gene expression was switched on to an even greater extent during the 7-week retraining period. The enhanced expression of these muscle hypertrophy-related genes was highly correlated with the largest increases in lean mass observed at the end of the retraining period. So the augmented hypomethylation of muscle hypertrophy-related genes during the retraining period was an epigenetic modification that allowed for the production of new muscle proteins and greater gains in muscle mass upon secondary exposure to resistance training. This is one mechanism by which human skeletal muscle cells possess an epigenetic memory of earlier exposure to resistance training, exercise, and muscle hypertrophy through periods of detraining and retraining. It cannot be concluded from this study how long these epigenetic changes last, since the detraining period was only 7 weeks in length. The authors do speculate that it's possible these epigenetic modifications can persist for years to facilitate enhanced responsiveness to resistance training after very long layoffs. Activation of satellite cells is another mechanism that muscles may use to form muscle memory. The satellite cell was named due to its peripheral location between the basal lamina and sarcolemma of a muscle fiber. Remember that a muscle fiber is synonymous with a muscle cell. Satellite cells are usually in a dormant state, but they become activated following muscle injury and when muscle fibers are under conditions of mechanical stress, such as resistance training. The satellite cell response to a bout of resistance exercise lasts for many days, but effects peak approximately 3 to 4 days post-workout. Once activated, satellite cells proliferate, migrate, and fuse to muscle fibers to repair them or induce muscle hypertrophy. When satellite cells fuse to muscle fibers, they can also donate additional myonuclei to the fibers. The additional myonuclei increase a muscle's capacity to synthesize new contractile proteins, which leads to muscle hypertrophy. Now, how is muscle memory linked to muscle fibers that have acquired extra myonuclei following training? Well, a 2010 study of mice conducted by Brusgaard et al. Stated that previously untrained muscles acquire newly formed myonuclei by fusion of satellite cells, which precedes muscle hypertrophy. Subsequent detraining leads to atrophy, but no loss of myonuclei. The elevated number of myonuclei on muscle fibers that had experienced a hypertrophic episode would provide a mechanism for muscle memory, and this could explain the long-lasting effects of training and the ease with which previously trained individuals are more easily retrained. I hope you enjoyed this Fitness Facts video. Thanks for watching.